Hello, so this first part here is DNA transcription. Basically, the DNA is unzipped and then mRNA is made, as you can see right there. So then this next part is translation, and mRNA goes out of the nucleus and it goes into a ribosome. And then the ribosome makes polypeptides, which are turned into proteins. So here's DNA replication. And what happens here is the DNA is kind of opened up a little bit. And then the primase, or helicase is what does that. And then primase comes and it puts down primers all along, which show where the polymerase is going to start putting down the nucleotides. And then, yeah, as you can see there. So ligase comes and then seals it all up. So here's how soap and salt open up a cell. So they break the membrane. And here's us adding the soap salt mixture on the side, and the DNA pours out. And then the ethanol is added by the great Mr. Sharp. And then basically the ethanol causes the DNA to rise to the top, and you can just take it out. So an endonuclease is an enzyme that breaks down a nucleotide chain into two or more shorter chains by cleaving the bonds. As you can see there, it kind of cuts it. So polymerase chain reaction, basically what happens here is copies of a gene are made. So, this is just kind of the process of it here. And then in DNA electrophoresis, basically there's gel, and then there's a electrical current running through it. And since DNA is negatively charged, so DNA is negatively charged. The more BPs there are, those stay at the top more, and then the less go down. And for some reason, part of it, this video is cut out, but the person 1 with 10 BPs is at the bottom, and then the 40 and the 40 stay at the top. So that's how you can kind of see, you can pair people's DNA and see if they, there's crime scene DNA and there's the person's DNA. You can see that they're the same with this. Bye.